All right. Thank you very much for everyone to join us. Today we have a special speaker, Professor Trey Ideker from UC San Diego. He is a currently professor of Department of Medicine and also adjunct professor in bioengineering and computer science. So as probably everyone knows, he's developed the cytoscope, which is the most well-used biological network visualizer software, and also the analyzing networks. So the cytoscape itself has over 30,000 citations. Well, I checked this morning on Google Scholar, and Professor Ideker has over 80,000 citations, including everything. But let me introduce a little bit about his background. His background is really interdisciplinary. Professor Ideker received bachelor's and master's degree from MIT in electronical engineering and computer science. And then he did PhD in University of Washington, Seattle, in molecular biology. And his advisor at that time was Dr. Leroy Hood. He is a founder or father of systems biology, and he's a co-founder of Institute of Systems Biology in Seattle. So probably many of you have heard his name also. Then Professor Ideker joined UCSD as assistant professor in 2003, and then became full professor in 2010. His contribution is not only the software development of the cytoscape, but actually he used that system biology approach for various different important problems, including cancer, which probably he is going to talk about today, and also like epigenetic clocks. So actually contribution is really more from probably biology side, which is what computational biologists like us needs to learn. So not just the software, which helps everyone, but really the important biological problems are really approached very nicely by this computational methods. So with this cytoscape and this network analysis, he is also currently the director from NIGMS Center of National Resource for Network Biology. Okay, so he also received one of the most prestigious award for bioinformatics. This is the Overton Prize from ICSP. ICSP is the largest international bioinformatics community. So it was 2009. So his contribution has been really recognized from early stage. And then just influence is expanding now because the network analysis system biology is getting popular. But that is founded by him and other colleagues. Okay, with that, I guess we have an interesting discussion here. Professor Ideker, please.
Okay, well, thanks for the invitation to talk and for that very honorable introduction. I hope I can live up to it somewhat here. The talk title, Building the Mind of Cancer, as noted, is one application of a larger idea, but the idea here is can we build ultimately a complete AI ML system, if you want to call it that has complete understanding of cancer. Okay, what does that even mean? Would be how do you even define that problem? Would be the goal. So let me start with the early days of systems biology, because in fact, also as mentioned, everything that I'm going to talk about is really an extension of just that, of what people and Lee Hood had called systems biology back in the early days. And so and of course, many authors have defined systems biology in different papers, and all the definitions are more or less different. So just operationally, the definition I'm going to use today is one that Lee and I used in this early review of systems biology, and we said it's an approach to the study of a biological system by perturbing it systematically monitoring the responses of the system at multiple levels, and formulating network models, or just models, to describe the structure of the system and its response to perturbation. So more or less a reverse, what we would call reverse engineering of any system, basically. Now, if you look at the date this paper came out, 2001, there's another set of really important papers that came out in 2001 in biology, and that was the Human Genome Project was declared complete with 2 back-to-back papers on essentially the complete human genomes of those two projects. It turns out, in fact, that the human genome wasn't really complete. It depends what complete even means, and people still are actually finally now have a complete thread that represents the entire one entire human genome. But if you think about what that project had achieved even back then, it was essentially what you would call today a data sciences pipeline, an end-to-end pipeline for determining the linear sequence of letters of base 4, ACGs and Ts, that make up the 3 billion letters in one human, in the human species genome. And yes, there are differences, but those differences are really 1 in 1,000 of those letters, and 900, or roughly, and the other 999 are the same, and they are what define us as humans. The question systems biology is asking, having finished the human genome, is what about the rest of the cell? What about the rest of the tissue? After all, that DNA is just a thread in the nucleus, and there's the rest of nuclear structure, which is a Jetsons-like city of machines inside of machines flying around along filaments that take molecular cargoes from one part of the nucleus to the other. It is a really complex factory of stuff inside of each and every cell, and functionally, cells have incredibly complex behaviors. People in neuroscience, of course, love to study, speak about the brain as the ultimate complex system biologically, but as it very well may be, but the cell itself is an incredibly complex system that is only really still poorly understood. So with regards to a data sciences pipeline, which is the human genome, we determine this using a systematic, semi-automated, or one could now argue fully automated approach. So what does the systematic, fully automated approach start to look like for the rest of biology? That really is in my view, the essence of systems biology. And 20 years later, we are somewhere in the middle of meeting that challenge. We're nowhere near complete in our ability to push a button and systematically read out a map of the entire cell. Now, as with any biological question, there's 2 sub-questions here, Q1 and Q2. Q1 is the structural side. What is the structure? So, if you think about the human genome, it's a structure. Okay, it's the structure of ACGs, and it's the sequence. We are still trying to figure out the function of those ACGs and Ts, by the way. So we still don't know that, but we at least know the structure. So question 1 for cell biology or for tissue biology is the same. What does the structure of that object look like? And Q2 would be, how do we then begin to understand not just the structure, but build models that are also predictive of complex biological states and responses? So these are all very grand questions, I know, but it's the first slide of my talk. And so now what I'm going to do is burrow in, as I've alluded, the entire field of systems biology, and certainly of my career, it has been devoted to addressing these big questions. And now what I want to do for today's, for 45 minutes or so in what remains of today's talk, is talk about each of those 2 questions and give you sort of late breaking updates from work in the past year or 2 or 3. So again, question 1 is a cell structural question. How do we create an automated data science pipeline for mapping the structure of cells, and two, how do we begin to functionalize those structural maps to create predictive models. And as you'll see, we're like the rest of the world enamored by deep learning. And here the question will be, can we create deep neural networks that are not just functional models of responses, but are also structural models of the cell. So let me just get started with the structural part first. So the sort of recent late-breaking update from my lab on structure is to combine what are undoubtedly two parallel major data types in cell biology. In fact, one could argue the two major data types in cell biology, which up until recently had been considered to be totally separate. Whether or not someone had thought of combining them, I don't know, and if not, it was simply because these were two very different branches of biology. On the left, we have microscopy, so peering at images of cells in 2 dimensions, sometimes 3, but here I'm focusing on two-dimensional snapshots, fields of view of cells taken with confocal microscopes. And on the right-hand side, the complementary approach from biochemistry. So microscopy on the left, biochemistry on the right. And in biochemistry, you don't look at cells, you grind them up and you analyze extracts from those ground up samples in various ways. And the way that, just to start with that, the way that a sort of state-of-the-art way of studying cell components and proteins in biochemistry is with this approach called affinity purification mass spectrometry, or APMS. So let me now, that's the 80,000-foot description of these two branches. Let me now go to the 20,000-foot description. So and this now invokes a particular configuration of using imaging or microscopy on the left and biochemistry on the right. So often both of these two technologies are used to study proteins and, in fact, protein positions. Okay, so how do you study protein position using microscopy? Well, you tag each protein of interest, ultimately every protein in a human cell, and there's roughly 20,000 genes, each of them encode about, essentially first order one or more proteins. So we'll say 20,000 proteins, and each of those proteins can be interrogated with a fluorescent dye attached to an antibody against that protein. And so here the target protein is fluorescing in green. Now you also in modern microscopy, confocal microscopy instruments have typically 3 to 5 other fluorescent channels that you can simultaneously image. And this just gets into how many emission wavelengths can be detected simultaneously with the device. And so here we're also staining the nucleus in blue and the so-called endoplasmic reticulum in red, which you see here splayed out around the nucleus. Those so-called counter stains will always remain the same from target protein to target protein. So we'll ultimately have 20,000 different target proteins fluorescing in green, always against the same blue and red. And it turns out sometimes a few other markers that'll stain other cell structures like cell membrane, for instance. And those will always be the same, okay? On the other hand, for biochemistry with APMS, we also have an antibody against our target protein. Here that in the biochemical lingo, that target is called a bait. And we pull down that bait from, again, a ground up cell mixture now. You're not preserving the 2D location and positions of cells, you're grinding them up. But as you isolate that bait, you also will isolate what other proteins that directly stick to that bait inside of cells, okay? And those are called preys or interacting proteins here. To identify those, you then use this mass spectrometry device. which will separate out those proteins by their mass, or more correctly, by their mass over charge. And then you can, through further fractionation steps, can identify what those proteins actually are for each of them. And that's a standard mass spectrometry modern setup. Lots of computational problems, by the way, in both of these that I'm going to gloss completely over, underlying both of these pipelines, okay, that are supporting 20-year research plans that develop these pipelines. In the case of this APMS, you're also positioning proteins simply relative not to cell landmarks like nucleus ER. You're positioning proteins relative to each other in some, but in some other space, okay? And then like my lab has done with Cytoscape, you can then draw these, or using software, I should say, like Cytoscape, you can then draw these networks of interacting proteins. But again, devoid of any imaging or any location notion of where those proteins are located relative to the cell itself. Nonetheless, both of these technologies are mapping protein position, just in different ways. And that really was the key insight of a student of mine, Yue Chen, who just published this paper end of 2021, which integrates those two pipelines together to start to get draft maps of cell structure. And here is how it's going to go. I should also mention this was a very enjoyable collaboration with the two groups who are, or two of the several groups out there who are leading protein imaging versus protein mass spectrometry. So Emma Lundberg is one of the founders of something called the Human Cell Atlas, which is part of the Human Protein Atlas project, which is in fact has measured already the protein positions inside at least one cell type for most human proteins. And then on the biochemistry side, we collaborated with Steve Gigi, Wade Harper, and Ed Hutland, who run what's called the Bioplex project. And this is the same thing running through each protein, but now using mass spectrometry to identify what are the neighbors of each of those proteins. Unfortunately, because these projects had not, no one had sort of proposed putting together these things before. These projects had run in different cell environments and cell types by and large. And we thought it was very important to actually not mix and match different cellular states. And so we ended up settling on a smaller project as a proof of concept, where we did have from both projects measurements in the exact same cellular cell type, the human embryonic kidney cell HEC293. It's a cell, and HEC293 cells are a workhorse cell type for lots of people. They grow really easily in the lab, and have lots of libraries of lots of assays that have been developed that use these cells. So everything I'm going to show you is only on 700 proteins. It's really a dry run for what we want to do next, and haven't published, certainly, or in the middle of doing, which would be to look at all 20,000 to really put in the experimental effort to synchronize these two pipelines for the same cell type, and then see how fast we can do it. Okay, so this I'm going to do it slow in 700 proteins. The challenge for 2022 and 2023 is, can we do it incredibly? Can we speed up this whole thing by 100x? Okay, but how does it work? So we simply so the answer is in terms of the computational aspects, it's simply using almost off-the-shelf componentry. So I think many on the call will be familiar with convolutional neural networks, especially for analysis of imaging. And so here we're using something called ImageNet, which had already been shown to work well in analysis of proteins in cells. After having been trained on many different kinds of images generally available on the web. And so here we're taking the penultimate, the next to last layer of the deep neural network that represents ImageNet, it has exactly 10,024 features. Okay, so essentially we're going to embed each of these images of a protein in 1024 features. Now think about the dimensionality of the input. I forget the exact number of pixels, but let's say it's 1,000 by 1,000 pixels. Then there's per image. There's a million dimensions, even if I don't talk about color and the number of bits I have in each pixel. So these are obviously very, very high dimensional data that I'm going to embed, compress down to 1,000 dimensions. And I should also talk about, typically I'll have 2 to 5 separate antibodies against the protein, which gives me some redundancy. So it's not just 700 images here, it's more like 700 times somewhere between 2 and 5. And then if I have replicates, another factor of 2 or so. Okay, and then it turns out I can do the same thing or a similar operation with deep neural networks to embed proteins based on their neighborhoods in protein interaction networks. And this also is an off-the-shelf component out of Yuri Leshkovich's lab. Some of you may have used something called Node2Vec before out of the Leshkovich lab. And it's very, very much inspired by the Word2Vec algorithm, except this traces paths, local paths through a node in the network instead of looking at sequences of words and sentences. But it's the same idea. And so with Node2Vec, I again will embed every protein, and we chose it to be the same number. You can tune with this algorithm how many features you want in your embedded space, and we chose also 1,024 features here to match the image net. One way to think intuitively about what happens here is 2 proteins get embedded by Node2Vec in a similar space. Yes, if they interact directly, but actually what's more important is that their neighborhoods are the same. Okay, is the way to think about it. So if two proteins have the exact or very similar sets of protein neighbors in the network, first neighbors, then they will end up in a similar position in the embedding, essentially regardless of whether they directly interact. Now, the final step here, which I'm talking too long, so I'm gonna gloss over, but just you can read about it in the paper, is essentially what amounts to a co-embedding. And by the way, it turns out there's some details that need to be better worked out. And so if you're looking for where's the weak point of this whole paper, I'll tell you right where it is, and it's right here in how we do this co-embedding. It's okay what we do. It's not optimal. I'll just tell you that right now. That said, what we arrive at is a single embedding where now so the points are proteins, and proximity between 2 proteins is now informed by both pipelines of data. That's the key. So 2 proteins are close, essentially, if they're close in image space and they're close in APMS space. Now, here I'm using the UMAP approach to essentially reduce further to 1,000 to 24-dimensional embedding, just so I can visualize it. But we don't stop there. And again, I'm going to gloss over this detail. And here, this is a detail where I think we've got it exactly right. And most of the time when you see an embedding, the position or the distance in the embedding doesn't really have a unit. It's some abstract thing. But here it turns out we were able to calibrate distance in the embedding between 2 proteins into something that looks a lot like nanometer distance of that component, of their shared component, or I should say nanometer diameter of the smallest shared component in a cell containing that protein pair. That's a precise statement, but that's exactly what it is. So I can sort of measure in this embedding and calibrate it to meter distance inside of cells. And the way that happens is by use of standards, where I have from structural biology experiments using technologies like X-ray crystallography, I have a number of gold standard structures in cells where I know how far apart 2 proteins are. Okay, but what do we do? To move from that embedding to something that starts to look like a cell structure, or as you'll see here, at least an ontology. What I really mean here, as far as we push towards cell structure, is an ontology of discrete cellular subcomponents we can find evidence of in the embedding. Okay, so we get an ontology of cell parts. But look, if I have to like make up some cartoon where I visualize this, where I project this ontology into 3 dimensions and fly around it and to show the cell structure, I can do that, but it's largely a fiction. The real data structure here is an ontology, but it's a data-driven ontology constructed directly from those 2 imaging and mass spectrometry data types. But to get towards that ontology of parts, I have to apply some kind of clustering approach in that embedding. And depending on whether you talk to computer scientists or physicists, this will be called clustering or community detection. And so here we're using an algorithm from physics. So I'll call it a multi-scale community detection. And so the idea here, let me start at the bottom left. Is you want to find modular protein communities where modularity has a precise definition from physics, which has to do with whether or not the density of edges inside of that group of proteins is greater than the density of edges connecting any one of those proteins to outside proteins. Outside the module, okay? So you're looking for essentially dense subgraphs of proteins, and the multi-scale part relates to the fact I can apply this community detection or clustering procedure at different scales or distance resolutions. So intuitively, if I'm interested in very, very short distances from say 1 to 15 nanometers in this embedding, remember I've calibrated the embedding, then I would find a cluster in this region of the embedding that includes these 4 proteins. Gemin7 is one of them. If I now repeat that clustering operation, but now using a bigger neighborhood, say from all the way up until around 20 nanometers, then I would, of course, add, or I could, of course, add some more proteins, and I've done that here with like CCDC12. And now, as I continue to expand the scale or the resolution of my clustering, now up at 200 nanometers, I find a bigger cluster still. And look, what's happened is that bigger cluster is really the merging of 2 separate clusters at the lower scale down. So what that, of course, gives me is a hierarchy of communities of proteins inside of cells, where as I move up the hierarchy, I'm essentially moving up in the nanometer scale or resolution of the analysis. And that, of course, those operations are going on simultaneously across the entire set of 700 embedded proteins, producing a hierarchy like the one you see on the right. We call this thing the multi-scale integrated cell, or MUSIC. And the ladder over here on the right indicates, well, the first, just to be clear, the nodes now are not proteins, they're communities. As you see over here at left, it's the inner hierarchy here at left that is now being shown over on the right. So the nodes are communities, and now the communities at the bottom of the page, towards the bottom, are smaller ones. The communities towards the top are larger ones. And as we've shown at left, of course, by construction, smaller communities will nest inside of larger ones. And the size resolution at which a community is detected is shown here in the size ladder over here on the right. And this is the thing that I really am quite excited about, because you don't usually get these kinds of direct distance calibrations for these hierarchies. And working down from top down, of course, the degenerate largest community is all proteins, and that essentially we call the cell. And then as I begin to factor that in a divisive way, moving down, if I want to think about it that way, I first get large organelles like nucleus and cytoplasm, and then I start to factor those organelles, such as nucleus factors into nuclear lumen and nucleolus. These are well known to biology. And then down here at the very bottom, I'm looking at very specific protein complexes that live inside of each of those large organelles. Do I find, and as you'll see here in a second, I find I recover lots of known biology that's in orange, and the blue or purple concepts or communities here have some novelty to them, and exactly how you classify the novelty gets interesting. In fact, how you label this ontology gets interesting. We do it by ontology alignment against the literature curated version of this, which some of you may know is called the gene ontology. Okay, but that's curated from literature. This is de novo generated from data. But first, just to convince you I can recover known biology, here on the left are 9 components in cells that were not used for calibration of this pipeline, but nonetheless are known in size and structure from structural biology studies in the literature and record entries in the Protein Data Bank, or PDB. The diameter at which that component forms in the music map is shown on the x-axis. The quote unquote actual gold standard, or at least bronze standard, diameter of that component from PDB is shown on the y-axis, and you can see over whatever 4 logs, we're getting a pretty good diagonal, a line which indicates a good recovery of these components. Although of course on a log scale, that'll certainly compress some errors. But here's an example of one, to pick out the pre-catalytic spliceosome here. Here's what the structure, as is known from structural biology, looks like. The colored parts of the structure are the proteins that are in our modeling exercise here. Remember, we only had 700. And so we get typically a subsampling, almost a random subsampling of any of these components. And those are the ones shown here in the colored parts of the structure, and the gray is not our proteins, not yet in our model. The estimated diameter of the precatalytic spliceosome by the data is 48 nanometers, and the measurement is 42 nanometers from literature. We can also put error bounds using an approach that is essentially, it's very similar to statistical bootstrapping. We can use that to where we subsample the input data to look at how much variability one then gets in our estimates, and so a reasonable size range we think for our estimate is between 26 and 90 nanometers, which of course contains the measurement of 42 nanometers. Now that's of course just 9 components that are well known, and I mentioned there's a lot of novelty in this thing, and so I think in interest of getting to some other stuff, I won't go over this example, but here's an example of a protein, RPS3A, that landed with what otherwise looked like a complex, a 4-protein community, which we call the RNA Splicing Complex, because the other members are known in RNA splicing, and through further experiments, we were able to show that RPS3A probably is itself indeed a splicing factor. So that's the basic first part of my talk on a new pipeline we have that really just realizes that 2 old, very mainstays, old but also current mainstays of cell biology, imaging or microscopy versus biochemistry, can be put together using modern techniques from modern ideas from machine learning. What I now want to show you quickly is just is that we're now trying to apply that to cancer cells and to scale it up from 700 proteins to a larger number of proteins by doing new de novo affinity purification mass spectrometry and then ultimately imaging. But it's going to take us a few years. And so here is sort of the ongoing results from the cancer mapping project, where we with our collaborators in the Nevin Krogan lab up at UC San Francisco have begun developing these large APMS networks for cancer cells, and run clustering / community detection in them to build these hierarchies of cell components. But now, of course, we can look at how those components differ between cancer and normal cells. And also we can look at which are the components in those hierarchies under mutational pressure in tumors. And so here is what, now I should also say we have not done any imaging yet. So this is simply based on the mass spec side. It's not based on any imaging. So I expect it's going to really focus when you add the imaging data, and we're working with Emma Lundberg to do that right now. But again, it's going to be a couple of years. Nonetheless, we've kind of tried to race ahead and look at what you would do next, which is you would then look at cancer genomes and the mutation patterns you see in cancer genomes to figure out what are the components inside cells which are under mutational pressure, or enriched, I should say, for mutations. And so this is what that component diagram or ontology looks like. This first author, Zhang Fan, thought essentially upside down from the way Yue thinks. And so when she puts the smallest components at bottom, he puts the smallest components at top, because he thinks about mutations kind of hitting these small components like rain, or like one of these Japanese pinball machines where you drop the balls in and then they kind of all end up kind of funneling. So if the balls are basically mutations, you want to see where do the mutations tend to embed in terms of components inside of cells. And there is some linear algebra that we think is quite nice that sits behind that for figuring that out. Where is the mutation pressure inside this map? Once again, I'll say I think there's room for improvement there. If you're looking for interesting projects, you know, how do you take structural biology knowledge like this and really understand how that intersects with genetic mutations? I think it has already, you know, is a very rich area. You know, here's one example, though, of where mutations embed in these communities as you use them to apply them to cancer. This is not even a new complex. It's we've essentially reconstituted a well-known machine inside of cells, or in this case, excreted by cells, which is collagen. Collagen is in our beauty. You know, you put on face cream, it's got collagen in it. Okay, collagen normally is the dominant fabric of the space between cells, what's called the extracellular matrix, or ECM. Okay, so cells excrete it, and it sort of stabilizes the tissue. Of course, tumor would need to has to push against collagen fibers as it expands, and then ultimately, if your tumor metastasizes and moves somewhere else in your body, it's going to have to break out of that collagen entirely. So it makes perfect sense that collagen would be mutated. But because it's a big machine and it's not a single mutation or a single complex, but there's hundreds of mutations that hit in different ways on that machine, all with the same effect of disabling it, that it had not been found before to be mutated in cancer. Okay, it had been studied in cancer, but had not been realized it was mutated. And so, and again, it's because you have to sort of take the machine systems view to realize that there's again, every patient mutates a different part of collagen. And so you're not going to find any common feature to those patients if you're looking at the nucleotide level, if your features are nucleotides, and even if your features are genes. Here, there are, 12 different, or was it 11 different collagen genes we're showing, each of which have some mutation frequency. In this example, this is lung cancer, okay? Half of lung cancer patients, 49%, are mutating this collagen complex, but they do so largely through different genes here, making it hard to realize or discover unless you know about the wiring diagram of the system. And then over here at right, we worked with Stephanie Fraley, who's a bioengineer who looks at this collagen fibers, and she was able to show that if we actually create, using CRISPR, we create some of these mutations in the matrix, what does it do to cell growth? And you can see here that cells, the Ki67 marker is a marker of cell growth, and the fact that these cells down here in the mutant matrix are lighting up like gangbusters just shows that there's something about that reprogrammed collagen fiber that is allowing cells to grow better. Exactly how it does that remains to be seen. Okay, so in the last few minutes now, what I want to do is turn from structure to function. So now we can begin to map, at least, if not like 30, 40 structures of cells, we can at least begin to chart out the ontologies of components, of discrete components inside of cells. Okay. How do we functionalize those? And I think that question can be answered in a lot of different ways. And also, why do we want to functionalize them? And I think I'll skip this slide because I've already said why you might want to functionalize it. You want to predict what's going to happen when mutations hit that thing, you know, and how does that govern the state of your tumor and what drugs that tumor will respond to. Okay, so when you talk about function, often in biology, you're talking about what could generally has been called the genotype-phenotype problem. So the genotype-phenotype problem is certainly function in genetics, I should say. There we go. Function in genetics really all boils down to the genotype-phenotype problem. You have an input genome, and you want to be able to predict observable phenotypes. The phenotype is simply a word just things you can easily observe about the organism, like do they have cancer or not, okay, or what drugs will work on that patient, okay, in the precision medicine context. Okay, that's probably the most interesting thing you can predict, that you can observe about a patient. Are they getting better or worse, right, given your current therapy, right? So these are all this is an instance of genotype-phenotype prediction. Okay, that's a functional problem, not a structural problem. But nonetheless, let's think about what is the structure. How does that relate to structure? Okay, well, what's the structure of a genotype feature? Well, a mutation, a letter of DNA. If an A switches to a T, I'm going to claim that's about a nanometer. That's an event on a nanometer scale. The scale of that event is a nanometer. Okay, you can argue maybe it's a tenth of a nanometer, but you know it's around there. What's the scale of phenotype? Well, if I say that patient is responding to drug, that patient is a meter in size, right, or at least order of meter in size, and this then leaves 9 or 10 orders of magnitude of physical scale between the genotype and phenotype. So in a sense, this functional prediction problem is also one that inherently involves bridging scales, mini scales, okay? And again, it gets back to one nice model of that is if you can imagine changes in genotype are now falling down like pinballs in my previous example, or like rain is another good example. These are funnels, and they're raining. Changes in your genome are raining down on this thing and embedding that, or getting funneled through different components that they affect. And as you move further down, the components, of course, these are nested complex machines, and so you're talking about the bigger machines that contain the smaller machines that were impacted, ultimately impacting the human machine. Okay, and how can knowledge then, as I've just described, that starts to look a lot like that ontology we just built for cells. How might that help? Well, as we just saw with collagen, it helps in the following way, shown on the bottom right. If I have rare events at the genotype scale, if those are my fundamental input features, it may be that I never observe a given feature again. I may have one patient sample that shows that feature as a mutation and never again. So it's very hard to do learning when you have an N of 1 sample, when you don't have a recurrent event. But if I now look at an internal machine inside of this hierarchy, I can start to put features together and observe that, okay, in some samples, A is mutated, in some samples, B is mutated, and so on. But if A, B, C, and D are genes that come together in a, or gene products, proteins, that come together in a machine, I can make a statement that many people are, or many samples are mutated in this machine. And that is as we saw with collagen. And so that's sort of the principle here. And I think how you implement this, there is a lot of debate, and I make no claims that what I'm about to show you is the right way or the best way, but it's certainly an interesting way that we have managed to couple that structural intuition to learning, a genotype-phenotype learning. And as I said, this is all in the context of precision medicine, where we give you a tumor profile. So what are the mutated genes in that tumor? We give you a drug that we are considering giving the patient, and we want the machine to predict, or this machine learning system to predict a response. But we're going to importantly constrain the learning to respect these hierarchical cell biological models, these systems biology models I've just been talking about. And this is another sort of series of publications that I would point you to. And of course, this is my talk, so I'll show the ones coming out of my lab, but there's been some others out there that are also very interesting. So here's how it works. It's quite simple. It's probably too simple. In these ontologies that we've been drawing now, okay, or determining from mass spectrometry and now imaging, each of those components will have an assigned bank of artificial neurons, okay? So the component that corresponds to, say, DNA repair, that module will have, say, these inner circles are meant to represent neuron states of the neurons assigned explicitly to DNA repair. We only allow non-zero weights that update those states to come from the subsystems nested inside of that parent system. So in this case, you'd have communities. DNA repair, it turns out, in those maps factors into multiple subcommunities, and those correspond to different ways of repairing your DNA. Double-strand break repair, base excision repair, mismatch repair are 3 of those ways. Each of those has a bank of neurons representing its state, and those are the only neurons that are allowed to update the state of DNA repair during learning. Now note, I'm going to now apply, or Zhenzu Ma, who was a faculty at your institution, I think. I'm sorry he's no longer a faculty there, but that's out of my hands. But Zhenzu Ma actually has a beautiful series of papers. And this is what he really came up with when he was in my lab, for pioneering how you do this. And he just used conventional neural, deep neural networks to train with back propagation over a series of epochs. And again, the learning here, even though you constrained it, the learning still happens because all these non-zero weights still get tuned, right? Now, what are the training data? He has a series of papers. I'm going to where there's different ways of, or different genotype phenotype training sets. The one that we've really, the real target here are these cell screens for cancer cells responding to drugs. Out there, you might be familiar with studies from the Broad Institute or from the Sanger Center, where they will expose up to about 1,000 different tumor cell types, each to about 1,000 different drugs. So you literally have 1,000 by 1,000 matrix, where each cell of that matrix is a quantitative, is a scalar basically, which is how dead the cell is, essentially how fast or dead it is growing under that drug. And you know the genotypes, you know the mutations in all those 1,000 cell lines. And so that's your input. You're trying to find what are the cell genotypes that respond that predict a high growth versus low growth in response to the drugs. I want to save time for questions, so let me. Predictively, this thing works no worse than the state of the art. Let me leave it at that. I don't need it to work better. You can argue there might be sometimes it works better. I don't want to get into that. It works. Let's just say it does no evil, does no harm, because the real target here isn't better predictive accuracy, it's interpretability. Now what I can do is now for a drug where I can explain, I'm pretty good. It turns out about a third of the drugs, the models do pretty well at capturing whether a cell is going to respond or not, given its genome. Okay. And now the question is why? Okay. So and it turns out interpretation, as you guys probably already appreciate, is a hard problem. It's even hard it turns out having assigned neurons to concepts, but it's a little easier. And so here's an example. Paclitaxel is a first-line therapy in breast cancer. It is thought to work by binding microtubules. So that's the machine they should interfere with. It turns out that in our model, microtubules the neurons representing microtubules are when you simulate, having learned the response, you then forward simulate the response, and you essentially look at what are there's different ways of doing it, but to a first order, you look at what are the response neurons, and microtubules are responsive to paclitaxel, but you also get glucose metabolism, and that was a new striking discovery. So complex or protein communities involved in response to glucose, cyclic AMP, insulin secretion related to glucose. All of these things came up as important systems in our model, which now can these are essentially, so the model is predicting essentially two things now. One, what's the response to Taxol, but two, what are the underlying molecular mechanisms that enable that response as a second prediction, and that second prediction can be validated. And so there's more in this cancer cell paper where this comes from, but here's an example. So if glucose pathways are really important for paclitaxel response, what happens if we perturb the system twice, once with paclitaxel and once to a directed perturbation to glucose metabolism with this glucose mimetic called 2DG, 2-deoxyglucose. Okay, it's not glucose, but it essentially gums up glycolytic metabolism. And you can see here in the third column, when I combine the two agents, I get this striking synergistic effect on cell killing. So just to wrap it up, what I've shown you today is sort of what I would call structural and functional systems biology, at least the state of that in my lab. I'm excited about this recent ability the UA Chin in my lab showed for combining microscopy and mass spectrometry proteomics to map cell structure, to give these data-driven ontologies of cell components. And then I'm excited that knowledge could have bearing on interpretable machine learning systems for precision medicine. And I think there's a really interesting discussion there into how that learning happens, and whether given enough training examples, maybe you don't need that structural knowledge. Given that there's 7 billion people on the planet, that's your upper bound of training samples. So one wonders whether you're going to have to also feed it some structural knowledge. So anyway, thank you very much. And if we have time for questions, I'll be happy to take some.
Hey, all right, thank you very much. Do you any questions? I see 2 questions on chat box. So the 2 questions are specific for the multi-scale integrated cell slides, music slides, and okay, so the first quick question was that, so in that music slide, you had a purple and yellow color code in nodes, and I think you mentioned it. What was the meaning of those 2 colors?
Yeah, I just quickly glossed over that. Those were the new versus established components that we had sort of read it. So which components have been rediscovered versus had some novelty.
Yeah, so second question I see in the chat box is about the how to interpret the large nanometer distance. So if the distance is very small, I think that means really physical interaction between proteins. And when the distance is a bit larger, beyond that complex, the physical interaction level, what would that really mean?
Yeah. So remember, when I embed two proteins, it doesn't necessarily mean they directly interact. It simply means it indicates their proximity in the protein network. And so if the two proteins, so as I move farther apart, it just means these two proteins may not directly interact, but they interact with a lot of common neighbors. And then, of course, as I continue to move farther apart, maybe it's neighbors of neighbors, and so on and so forth. So maybe that helps. And the imaging is the same. I mean, with imaging, it just means the two images look exactly the same in terms of where that protein is. Both proteins are exactly in small nuclear puncta, right? And those speckles look to the ImageNet algorithm exactly the same versus increasingly different. So that's what it means. And it turns out, though, that those that distance, that type of distance, does correspond to the size of the smallest component that contains those two proteins. That was the sort of key calibration realization.
Okay, thank you. Any questions from the audience?
So there's one that just came from Charles.
Okay. Okay.
Yeah, I don't know. I have no idea. Well, I would even say, is there to what degree is our music map fractal? Might be the first question. Is there sort of scale-free? Is there a scale-free property there? I don't know the answer.
Okay. All right. I have a general question. So here, you developed many interesting tools. Now everything is kind of connected in hierarchical fashion from the protein interaction scale into the individual person scale. And also from ontology level, you connected nicely mutation to the phenotypes, and so there are lots of work done here. So what do you still see something is lacking in systems biology? What else needs to be done to have, I don't know, comprehensive or I don't know, predictable building predictable or like comprehensive?
Well, so I would sort of double down on the framework. I feel most confident that I think the framework is an interesting, I mean, it certainly is a very general way of doing biology, right? Let's systematically determine the structure of the thing, and then we'll systematically fit functions into that structure, right? Or fit that structure into functions, right? But I think exactly how you do that is completely up for grabs. And I think, there's lots of ways, there's lots of room for improvement that both in sort of near-term and long-term projects. I mean, near-term, it's things like, here, the input feature vector to in Ma's work, the input feature vector is just a list of genes, and each gene has a binary state, 1 if mutated, 0 if not mutated. But we know that mutations, they happen not to genes, they happen to nucleotides, or larger than genes, alterations can happen to the tumor genome, which is large chromosome arms, can change together. And so the resolution of our input feature vector is both too small and too large, depending on, so there's lots of work that could be done in looking at the input. Also, how does one actually determine what are the important systems in this visible or interpretable neural network, I think is fairly up for grabs. And even harder, how does one experimentally validate those predictions? Right. When I tell you, DNA repair is an important process to these cell line, responses. How do I, those are all still hidden, very, they're visible in the sense that they correspond to an actual biological component, but the states are very much still hidden in terms of what we can observe inside of cells. And in terms of biology, there's certainly markers of some of those components. So DNA repair, over 50 years of research has produced, a number of very specific markers of DNA repair at the molecular level. But what we need is something much more general and systematic. So maybe gene expression data might hold some of that, future. Anyway, longer discussion, but yeah.
Yeah, thank you. Majid, do you have any questions?
Yeah, wonderful talk. Thanks, Trey. So I have talked with John when he was here, and then we had a lot of fun discussion about all the work. So, what is the contribution of each of these components? For example, you had confocal images gone with the mass spec data to eventually calibrate how close two proteins are. If you didn't have one of these data, or if you were replacing, let's say, confocal images with transcriptomic data, do you think that any of those would have been calibrated or addition?
Yeah, so we did. So in terms, I think the general question that you're asking is what's the relative importance of each of the data types, and how does that vary as a function of scale? Would be one. Well, I've rephrased your question into the question we actually answered in the paper. Let me put it that way. So you can see, so in the paper, we did look at that. And as you might expect, you see that lower in the hierarchy where the smaller scale components are, those are more informed from affinity purification. So you're the immediate small neighborhood of a protein. Whereas imaging really kicks in at the meso and large scales in that model. And it's not too surprising, right? Because imaging, people know a mitochondrion. I can rediscover a mitochondrion as a protein community of size 1,000 proteins, right? There's about 1,000, right? It's a massive clique-looking structure in the protein network in cytoscape, if you look, right? Is that the best way to discover a mitochondrion? No. You look under a microscope and there it is, right? So microscopy is really good at those upper organelles, but actually surprisingly, especially with ImageNet is surprisingly good at even though your eye can't tell that these two proteins are in the same complex. You might have 100 proteins whose locations in the image look very similar to your eye, but ImageNet pulls it apart into 3 or 4 complexes. So anyway, so that answers part of your question, I guess.
Yeah, that absolutely does. And do you think that if you had like special transcriptomic, like the transcriptome,
exactly,
that would.
Here's an example of future. Exactly. So I think that'd be very interesting.
Cool.
Okay. So can you answer the last question? It's on chat box. So Shwetal is asking if chromosomal accessibility and epigenetic changes can.
No, I mean this is exactly like we were just. I mean, so I can absolutely imagine that the ATAC-seq and methylation profiling would have a lot to do with with the modeling I presented, and thinking about how to actually incorporate that, I think would be really interesting. We haven't done it.
Okay, so let's thank the Professor Ideker again. It was really a pleasure to have you here. It was a great honor for us to have you really here. Thank you very much for a very nice,
thank you guys.
exciting talk.
Bye.
Wonderful. Thank you.